Good afternoon, everyone. My name is Eric Green, Director of the National Human Genome Research Institute, and it's a pleasure for me to introduce today's wall speaker, Dr. David Goldstein. David's biography includes earning a PhD from Stanford University in 1994, and following postdoctoral training, he took a faculty position first at the University College London as the Wolfson Professor of Genetics, a position he held from 1999 to 2005. He was then recruited to Duke University as the director of the Center for Human Genome Variation and also professor of molecular genetics and microbiology and biology. And last year he became the Richard and Pat Johnson Distinguished University Professor at Duke. David has many awards, including one of 7 Royal Society Wolfson Research Merit Awards in the United Kingdom for his work in human population genetics. And most recently, he was elected the co-chair for 2011 and the chair of 2012 of the Gordon Research Conference on Human Genetics and Genomics. And David's a very talented and insightful genomics researcher, which is why we brought him here for a Walls lecture. He's widely regarded as a leader who is critically thinking about how studying advances in technology, specifically advances in whole genome sequencing approaches, are going to really be used for identifying the genetic basis of common diseases and also drug responses. And as many of you know, genome-wide association studies, also known as GWASs, allow researchers to zero in on genomic regions that may harbor variants conferring risk for disease. But many questions remain about whether such studies can find all risk-conferring variants, and if not, how those undetected variants are ultimately going to be identified. And David's really been at the forefront of this debate and these investigations, and has really involved his own work to try to dissect this problem, specifically and most recently using next-generation DNA sequencing technologies and new analytical approaches for studying really a number of important diseases. And he's going to describe some of this work today in his talk that's entitled Human Genetics and the Next Generation, Rare Variants and Common Cures. David.
Well, Eric, thank you very much for that introduction, and it really is a privilege for me to be here today. I will be talking about the role of both rare variants and common variants in traits of medical interest, and at the end try to make some commentary about the direction I see that research going in the future. But before launching into too much detail on the rare variant side, I want to just kick off with some discussion of some of the work that we've been involved in looking at the role of common variants in conditions of clinical importance. And for anyone here to see sort of the latest chapter in the GWAS wars, I'm afraid you might be a little bit disappointed because I have some rather positive things to say about genome-wide association studies and some rather at least concerning things to say about sequencing in some regards, but let's just launch into it. So the first work I'd like to tell you about is studies that we've been doing to look at the genetic contributions of variable response to treatment for chronic infection with hepatitis C. And the background to this work is really clear, I think, to anybody interested in the genetic control of clinically important traits. If you look at what happens when standard of care treatment is used for chronic hepatitis C infection, you see that the standard of care treatment is really very far from optimal. First, at a fundamental level, it only works about half of the time. So about half of the time that patients go through the full course of treatment, the virus is gone for good. So you've achieved what you set out to do. It only works about half the time. On top of that, the full course of treatment is really quite arduous. It's always unpleasant, and it is sometimes dangerous. And finally, there has long been a quite strong indicator that there is a genetic contribution to this variable outcome to standard of care, and that is that individuals of European ancestry are known to be much more likely to be cured by standard of care than individuals of African ancestry. So all of these things made us very interested in looking at the genetics, and as I will comment more in a little bit, if you're interested in the genetics of a trait like this, then it is the case, even for somebody like me, that the first thing that you do for sure is carry out a genome-wide association study, which is what we did. And when we carried out a genome-wide association study, we found a variant, that's the RS number, that was near a gene called IL-28B, which encodes, I'm going to tell you very little about that gene, but it encodes interferon lambda, which is a protein that is in fact related to interferon alpha, which is used in the standard of care treatment. And we found a variant that has a dramatic impact on what happens when patients are treated. So here you see in the different racial and ethnic groups, individuals that are broken up by genotype. Here's the good response CC genotype, homozygote, heterozygote, and the poor response TT. And what you see on the y-axis is simply the proportion of individuals in each of those genotypic groups who are cured, and that the lingo in the field is sustained virological response. And so what you see here is that for individuals that have this homozygote type, more than 80% of them will be cured by this treatment, whereas individuals with this TT genotype, only just over 30% of them will be cured by this treatment. And when you talk to clinicians that actually see these patients, most of them agree that this is a difference that they would want to talk to their patients about. The other thing that's really quite striking about this variant is the consistency of its effect across different racial and ethnic groups, where you always see the CC homozygotes doing much better. And you will recall that I mentioned that individuals of primarily African ancestry are known to do much worse. Well, what happens if you look specifically at individuals of African ancestry that have the good response homozygote genotype and compare them to individuals of European ancestry that have the poor response homozygote genotype? Turns out these individuals, in fact, do on average much better than these individuals. So this really strongly emphasizes the importance of zeroing in on the underlying genetic cause of these average differences between the racial and ethnic groups. It's being much more informative than just using the racial and ethnic label. And in fact, as people that don't know this story may already be guessing, the good response C allele is the dominant allele in individuals of European ancestry. It is the minority allele in individuals of African ancestry. And that allele frequency difference, in fact, explains more than half of the difference in average response rates between individuals of African and European ancestry. So this came out of a genome-wide association study, clearly the right tool for that particular problem, identifying a variant that I think has unambiguous clinical relevance, although I would also like to emphasize that there is most certainly not algorithmic clarity in what you ought to do clinically as a function of genotype. I think all you can really fairly say is that this information gets incorporated into a kind of overall gestalt evaluation of what ought to happen. So if you had an individual, for example, with no signs of liver damage at all and a poor response genotype, you might then delay treatment for newer options to come online. I don't know what that warning means. But it does mean I can't advance. Does anybody? Oh, here we are. Okay, so I should say I have a disclosure to make. So Duke and investigators at Duke are beneficiaries of a patent associated with the discovery. I want to make that disclosure, but I do want to point out that it's not just my perception that there's clinical relevance. The test is being offered by LabCorp. And at last count, there were 1,000 or 1,200 tests being ordered a month. So this is actually being used in the clinic, and I hope in the end will prove to have been beneficial in that setting. We carried out, in fact, another genome-wide association study using the same data, and the data set I should mention came from a trial that was run by what was then Sharon Plow, called the IDEAL trial. This genome-wide association study was focused on one of the important adverse events associated with this treatment, and that is anemia. A majority of patients that undergo this standard of care therapy have a significant decline in hemoglobin levels, and that is due principally to the ribavirin component of the standard of care treatment. And so we asked whether there were gene variants that mediated the extent to which individuals suffered hemoglobin decline. So we simply took the decline in hemoglobin over the first 4 weeks of therapy, assigned that as a quantitative score to individuals, and then carried out a genome-wide association study. And then we found, again, a genome-wide significant association, this time on chromosome 20. And here there were a set of variants that went up towards 10 to the negative 50. So again, absolutely no question at all that we have a real association with who suffers anemia. But here's one interesting comment about interpreting GWAS. So all the variants on the GWAS chip that showed association were sitting in this open reading frame right here. So here are the variants that have this very strong association. You don't see association nearly so strong anywhere else. They're all sitting in that open reading frame. But we know for sure that open reading frame is innocent of any effect whatsoever on anemia. what's happening is that these variants are in fact markers we were able to work out for two functional variants in a neighboring gene, this gene ITPA, that encodes inosine triphosphatase. And there are two functional variants in the gene that are low activity variants. And it turns out that those two functional low activity variants are associated with these more common discovery variants. And they're not on the gene chip. So all that's happening is that these more common variants are reflecting the effect of those rarer functional variants. We were able to prove that statistically by doing the obvious thing, taking those two functional variants, genotyping them in the full cohort, and then asking, once you account for those two functional variants, does that make the initial genome-wide association signal go away completely? And the answer is yes, in every racial and ethnic group. So at a statistical genetic perspective, those are shown to be responsible. And we have, in fact, also been able to work out the biology behind how this is happening. I'm not going to go into that at all because that would be a little bit of a lengthy story. What it has to do with is how low activity of this enzyme allows the maintenance of ATP levels in red blood cells despite the effect of ribavirin on knocking down ATP levels. And basically, the ITP buildup that's caused by a reduction in this enzyme allows the biosynthesis of ATP to continue as before, despite the presence of ribavirin. So anyone who's interested in that can see the work that's just come out in gastroenterology that was done by a postdoc in the group, Yuki Hitomi, whose name is indicated there. And I think this is a really, to me, encouraging example of how you can get a pointer out of a genetic association study that really leads you to a biological characterization of what's happening. So I think that that really makes clear, and there are, I don't mean to say for a second that the examples only come from our work. There are many examples where genome-wide association studies have indeed found clinically useful findings and also insights to biology. But I think that it's also fair to say, and as I have argued, of course, before, that there are many traits where the variants that are responsible are really not very suitable to be detected by genome-wide association, and that's simply because they're too rare to be easily detected by genome-wide association approaches. Now, for some traits, and I think they actually have a pattern to them, the traits that are more subject to common variation, for some traits, genome-wide association approaches describe a lot of what's happening, but for a lot of traits, they won't. And of course, that has led to a tremendous amount of interest in developing the basic machinery that will enable us to use sequencing strategies to identify rarer variants that contribute to traits of interest. And I'd like to spend the bulk of the talk on talking about how we're approaching that right now and where I see that work going in the coming years. The work that I'm going to talk to you about depends upon the output of a small genome facility compared to the genome centers at any rate that we operate at Duke. The genome facility is run by Kevin Cheyana. It currently consists of 11 genome analyzer 2s, which are being phased out, and 6 HiSeqs, which are just being brought into operation. So I would say that that's small relative to genome centers and relatively large compared to places that aren't genome centers. I can certainly say that my institution has been very surprised at the cost of trying to store the data that we're generating, and there are ongoing discussions about how we actually do that. In any event, the data come out of this facility, and we have put a lot of energy specifically into 2 bioinformatic areas in order to work with these data. One thing that Dongliang Ge has done over the past few years is try to develop what you might consider a kind of Apple Macintosh type approach to looking at the variants that emerge from sequencing data. So I think there are a lot of ways that people are thinking about annotating the variants that are pretty consistent, but what we have found is that it's really important for users to be able to interact with their data, to be able to look up things in genes of interest and pathways of interest, and to interact in some reasonable time frame with the underlying sequence data. And so Dongliang Ge has set up what I think is an extremely easy to use user interface that has a kind of browser-like structure to it for looking at the variants that are identified in each of the genomes that you've sequenced and making comparisons between types of variants. So anyone's interested in reading about that software, it's available at the website that's indicated. And the other area where we've been pushing development a little bit, as opposed to just deploying the methods that others have developed, is in calling copy number variants using the data emerging from the next generation sequencing. And what we have found, as also using a quite different approach Evan Eichler group has found, is that whole genome sequence data really does afford a truly stunning resolution for defining the copy number variant landscape. So if you're talking about duplications or deletions of greater than, say, 2 kilobases in size, those can be hard down to that kind of size to pick up by alternative methods. But the read depth data emerging from next generation sequence data gives you really good resolution on those copy number variants. Now, the way that we have set this up, we actually make use of the alignments that come from BWA that you use for other variant calling. And Evan Eichler's approach is to use an alternative set of alignments. We have an approach that uses the BWA alignments that we think affords very good resolution, and it incorporates a specific novelty, which is it makes use of information about where heterozygote sites are called in the genome, and it basically precludes those from being called deleted, and that sort of grounds the calling and makes it perform, we think, better. But in any event, I think it's quite clear that you can get very good resolution about copy number variant variation in the genomes, and that really, I think, long term is going to constitute a very strong motivation for most of the sequencing being done by whole genome as opposed to whole exome. Not that you can't get copy number variant data out of whole exome data, but it's not going to be as high resolution, and so as the sequencing costs fall, I think we will primarily be turning that direction. So the first effort that we made in trying to see what kinds of things come out of large-scale sequencing studies was reported relatively recently in PLOS Genetics. And I only want to really emphasize one thing from this study, and that is just the sheer magnitude of genetic variation that emerges from these studies. I've got 2 plots here that illustrate that. What I'm showing here is the number of genomes that we consider, and on the y-axis, the number of novel single nucleotide variants. And what that means is that for the first genome, it's a variant that isn't in any databases. This is, of course, at the time that we did the analysis, it'd be a little different now. A variant that's not in any of the databases. And then the next one is a variant that is in this next genome, not in any of the databases and not in the previous fellow that you sequenced. Okay? And then so on. And the really striking thing is that after you've already sequenced 20 genomes, every time you sequence a genome, you're seeing more than 100,000 single nucleotide variants that you have not seen before. Now, for sure, some of these are not real, but the vast majority of these are real, and so it's a tremendous amount of variation every time. But more than that, there's a tremendous amount of functional variation. So we took all the variants that were predicted to be protein truncating and homozygote, and then we constructed the same analysis, same kind of analysis, and asked, how many new genes do you learn about that appear as being predicted to be carry a homozygote protein truncating variant? So you predict the protein's not made. And what you see is that even after you've done 20 genomes, you still get 3, 4, 5 such genes identified. Now, I do want to emphasize that we ourselves followed up some of these, and a proportion of these are not real. And on top of that, the proportion of these kinds of variants that are not real is higher than a random variant, because these are unlikely variants. And so if you actually see it's an unlikely variant, it's even more enriched for things that aren't real. But nonetheless, this shows that there is a tremendous amount of functional variation in every genome that you sequence. And that, in fact, is going to be one of our challenges in trying to identify pathogenic variants. You can't just look at a variant and say, oh, well, this does something quite dramatic to a gene. It must be my pathogenic variant. That certainly is not going to work. Okay, so will sequencing work? I want to give just a couple of examples that I hope will be on the encouraging side, that make some cautionary comments, and then back, I hope, to being encouraging. So, our own most recent study evaluating whether sequencing can work in the sense of finding pathogenic mutations that you have no information about before comes from a study we did of microcephaly. So this is work that was done in collaboration with colleagues from Israel, Elon Press in particular, and Doran Lancet, and run by Elizabeth Russo in my group. And the start of this was that in Tel Aviv, they had 2 families that both had a microcephaly syndrome that the clinician felt was a new syndrome, not described before, and the same in the two different families. And so naturally we wanted to know, well, in that situation, can you sequence and find the mutation that's responsible for the presumably same microcephaly syndrome? So here's the first family. So you can see that it looks like it's autosomal recessive. There are two affecteds that we sequenced. That's a fetus that was aborted on the presumption that the fetus would have the microcephaly. When we sequenced by whole exome sequencing these two individuals and then looked at all the variants that we found and compared them to control genomes, we only found 3 variants that were homozygote, functional, and homozygote in both of the affecteds, only 3. Okay? So in that family, we had it down from the exome sequencing to 3 candidates. And then we looked in the other family, and sure enough, one of those 3 variants is homozygote in that affected individual. And we then went on to type another 1,000 individuals for that variant and did not see the variant. Now, what I find particularly encouraging about this example is that we had it down to 3 candidates in the first family. And I think this makes a really strong case that even in isolated genetic conditions, you have some hope of finding the genetic cause. And I think that's a really important observation. Now, of course, we had to have this family to prove it, but it is down to 3 candidates in this first family. And being able to do that really did depend critically on the filter of absence in control genomes. And that's one of the reasons that this framework can work for high penetrant mutations. When you can actually filter that way, it makes it a great deal easier to find what's pathogenic. The gene that has the mutation in it has not been implicated in any microcephaly before, so this is a new insight into a cause of microcephaly. It's in the asparagine synthetase gene. And our first thought was that the mutation that's causal, which is highly conserved, as you can see there, our first thought was that the mutation would affect enzymatic activity. And so we set up an experimental assay to be able to test that. But then the postdoc who did that work found very quickly that the mutation, in fact, causes a profound instability of the protein. And so we couldn't even get enough protein to be able to carry out the assay. And so now we think what clearly is happening is that you just don't make very much of this protein when you carry the mutation, and that's what causes the microcephaly syndrome. And so now, of course, it will be very important to look at, see what the effect of this is in a mouse knockout. I have another perspective on asking the question of whether sequencing ought to work, and this is now not a real study, but this is kind of a Gedanken experiment. Let's return for a minute to the anemia example, where what we looked at was the quantitative change in hemoglobin for each of the subjects in the genome-wide association study that we carried out. So each of them have a score of the change in hemoglobin, and we can ask, would we have been able to easily find those causal mutations by sequencing if they, for example, had not shown up in the GWAS? And remember, we actually got very lucky in the genome-wide association study. They're not on the chips, and we only found the marker because 2 of the causal variants happened to associate just right with a common variant, so we found it. Maybe we hadn't have gotten so lucky, we could have tried it by sequencing. We can ask, would we find it? Well, let's set some kind of a significance threshold. Let's say that we're going to take all the non-synonymous variants that we see in the genomes that are present, say, in more than 2 individuals, and we're going to test for all of those variants. And so we have to correct for that. So call the significance level 10 to the negative 6, 10 to the negative 7. And let's say that your budget is finite, and so instead of sequencing everybody, you're going to sequence the ends of the distribution. So here's hemoglobin change for all the subjects in the study that we did, the GWAS study. Proportion of individuals in each of these, and let's say that you take the upper 5 percentile and the lower 5 percentile, so those with the most change in hemoglobin during the course of treatment and those with the least change of hemoglobin. Sequence these individuals, sequence these, and interestingly, if you did that by whole exome right now, the cost of doing that, 50 individuals of each, the cost of doing that is just over $100,000. Okay? So that is not an expensive experiment now. Sorry, the direct cost of reagents and labor to generate the variants right now, you still have to have people that can do something with the variants, but the direct reagent cost, excuse me. So what happens when you do that? Well, it turns out that the most important of those two functional variants that I described have a p-value of 10 to the negative 13 in a simple case control comparison between these individuals and these individuals. Very easily findable by sequencing, no question about it. Whole exome sequencing. And so here's an interesting comment. Had we not done a genome-wide association study for this trait, but instead jumped to hit it with sequencing, this would have been a complex trait that you actually can easily find the genetic cause for by sequencing. So I think that really is an encouraging example. It could almost make you wish that it wasn't discoverable by GWAS, but in any event, I think that it does make a case that it is certainly possible that some traits will in fact be amenable to discovery by whole exome sequencing complex traits. As far as I know, there isn't yet an example out there. Okay, so I'm going to now turn to the basic approach that we are using for complex trait discovery with sequencing. And I just want to give a little bit of the background to our thinking about that and then launch into a few of the studies that we're doing. So the first thing to really emphasize, which in fact is a bit different from genome-wide association, is just how important it is to be careful with who you decide to sequence. It remains expensive per individual compared to genome-wide association. And also, you're going to sometimes be making very fine distinctions amongst individuals. You want to be really sure of a phenotype in the individuals you sequence. So here are the kinds of designs that we're thinking about. One of the designs that we're really pushing is to take multiplex families for conditions of interest and to sequence some number of affecteds from those families. And depending on the information you have in the family, a good design might be, for example, taking distantly related co-affecteds, or if you have some linkage data, it might be just one individual. But sequencing within multiplex families allows you to leverage co-segregation data in those families in the analyses. And the other design that we are thinking a lot about is taking extremes of some kind from the population so that that enriches for causal variance, as you can see here, and then sequence those extremes, look for that enrichment of causal variance, but critically, take those variants back out into the larger population for confirmation of their association with the trait. So those are the two designs that we're trying to make the most use of, and I'll tell you about some examples of both of them. But before I move into that, let me just make some general comments about what I see as the relationship between genome-wide association going forward and sequencing. So the first comment is, if you have a new trait, I do take the view that it's very sensible to carry out a genome-wide association study first, because for some traits, it can be a quicker route to uncovering its genetic control. And I should just clarify for the record, for any trait that you're interested in, you would like there to be GWAS data already. So that means that it was a very valuable process to go through to generate the GWAS data. It's a separate question of how much GWAS data you actually need for any given trait. But whether you do a credible GWAS for a given trait, I think that that's really not something that reasonable people can disagree about. But for most conditions where there already has been a GWAS done of reasonable size, I don't think that GWAS practiced alone is going to be an important driver of discovery anymore. And the reason for that is that if you have a condition where you've got lots of signals already and you're having trouble tracking down to the causes of those signals, just adding a few more tens of signals to those conditions, I think, is not really where we want to concentrate our attention. We want to concentrate more on finding the causes of the signals. And so that means that I think we'll be interpreting the GWAS signals along with the sequence data to try to find the causes of the GWAS signals. I'd also like to emphasize that that combined use of GWAS signals and the sequence data is maybe particularly important given that GWAS is a very accurate and well understood experiment, and sequencing is not. And so one just has to really be absolutely clear about that. We're really good at doing genome-wide association studies, really, really good at it. So we've got the genotyping accuracy right there when it's done carefully, when the work is done carefully. We've got it right where we want it to be. We know what the artifacts are. We know how to control for population stratification. We know how to correct for the number of hypotheses that we're testing. So we really have worked that out well, and what that means is if you have a signal from a genome-wide association study, whatever you think about the value of it, it's a real signal when it's done right, and that's really important. And so I think that's one of the reasons that we want to think about doing them in tandem. And I also would say that without taking a view on what proportion of genome-wide association signals are synthetic in the sense that they are driven by relatively rare variants that just load up on the common ones without even taking a view on the proportion, I think it's pretty clear that some proportion of the GWAS signals must be due to this effect. And if so, they provide a good pointer for going in and looking for some of those higher impact rarer variants. So I think we really see, I see, the value of using these two types of approaches together. I'd also like to just emphasize a little bit from our own experience just how, I guess dangerous is the right word, sequencing is from the perspective of false positives. And I really want to make that point in that I think one of the unappreciated contributions of genome-wide association was to really clean up the literature and to reduce attention to a whole bunch of false positives that really did plague a lot of fields. There were some disciplines where the vast majority of genetic discoveries were simply untrue. And when you went and looked at the genome-wide association study carefully, that became pretty clear. And that represents a tremendous waste of effort when one is chasing these things that aren't real. And GWAS really did, I think, help to clarify what was real association and what wasn't. And GWAS simply represents a better association study than candidate gene studies in really every sense. And so I think it's really critical that we don't let sequencing go down that old route of being flooded with false positives, but it would be very easy to do. So here's an example where we're sequencing schizophrenia genomes, okay? And we bump into a mutation that introduces a premature stop in a dopamine receptor, okay? So we have a clearly, dramatically functional variant in a gene that is of clear relevance to schizophrenia by really just about anybody's definition. And could we have managed to publish that if we wanted to? You bet, you could publish that. But you also know that these kind of things happen. You sequence genomes and you get variants that have extreme effects. So we decide to sequence it before trying to publish it by other methods, Sanger sequence, and then we show first that we can actually see the variant in some more cases, but we also see it cropping up in controls. But more than that, we also, when we look at the next generation sequence data and the Sanger sequence data, we in fact at the end of the day can't be absolutely 100% positive that there's even a variant there, okay? And sometimes it can be difficult to determine. So at every stage you've got problems, genotyping accuracy and also storytelling from variants that really look good. So from my own perspective, I think that it's really all important that we establish a really powerful framework to establish what is convincing association and also to really prevent too much, there'll always be some, too much of the false positives coming out again in the literature. And so I guess what I'm really saying is that I actually perceive the possibility, maybe the likelihood that there's going to be a little bit of a Wild West mentality of people really forcing answers out of the sequence data any old way they can get them and making up a lot of stories. And like happened for GWAS, I would like to see the sequencing community come together a little bit and figure out how to do this right. So here's our thinking about how to do it right. The first comment to make is that you do not declare a discovery from the sequence data before you separately genotype the variants that you think you've made a discovery. Show that the genotyping is accurate and convincing with a separate technology. So the sequencing is discovery. You take the variants and you prove that you've actually accurately genotyped them. Now maybe in some future date, genomes will be sequenced so perfectly we can skip that, but I think maybe that's a little ways down the road. Here are the options for follow-up right now. They're actually pretty good. If you want to do some few variants that you're very interested in, I'll tell you about some we've done. You can just TACMAN them, and that's a separate technology that often is completely convincing, sometimes not. If you want to do some number of hundreds of variants, then an appropriate technology is Beat Express. And if you want to do some number of thousands of variants, an appropriate technology is iSelect. And I should also say that I do not own any Illumina shares, and I just happen to think this is a good approach for the follow-up. But there probably are other companies out there that could provide reagents for some of these. But finally, if your focus is on a gene, for example, there are multiple variants that appear associated as opposed to a single variant, then there is in fact a new sequencing technology coming out from Illumina MiSeq that is really exactly perfect for that, where you can take on the exonic regions of up to, say, 30 or 40 genes, so 400 exons in some number of hundreds or thousands of samples, and that cost might be $50 or $100 a sample. So this really, I think, is the follow-up framework. And I'm going to now tell you about a few studies we've done to carry out these follow-up studies. So the first one is taking a particular class of genetic variation and systematically taking all the variants that are identified for follow-up from that class of variation. And so we focused first on variants that introduce premature protein truncation. And the reason that we did that was twofold. One is that these are very clearly functional variants. And of course, the other reason is that we know for neuropsychiatric disease that copy number variants, which of course do the same thing but to multiple genes in heterozygote form, just reduce the expression, are associated with many neuropsychiatric diseases, and that maybe suggests that the mutations that are point mutations but that can knock down the expression of a gene might also be risk factors. So what we did is combine samples that we were sequencing in schizophrenia and epilepsy, making heavy use of samples generated with ARA support. And we looked at the sequence data that we generated and looked at all of the stop variants there, focusing on 114 genomes that we had sequenced with schizophrenia, 96 with epilepsy, compared to 150 controls, and all of the variants that introduced premature stops that were present in homozygote form we took for follow-up. Any variant that was in heterozygote form in 3 or more cases we took for follow-up. That was just a function of how much we could invest at the time. And then we also looked at special regions of the genome, for example, regions where there were copy number variants that had been implicated already in the condition. And we took all of those variants and put them into a beat express genotyping assay in a larger number of samples. And so that's a kind of intermediate screen to try to see which of the variants look interesting. So there were 384 stop gains that we were following up. These were genotyped then in more controls and more cases. And then we found in that follow-up that 16 were present in more than 2 cases and no controls. And the rest of them went away because we actually had found that they appeared in controls. And so now we're down from the 384 to 16, and we follow up those 16 now with the TACMAN approach, and we remain with 6 that are still invariant. Now, I don't know that we are actually zeroing in here on anything that's actually causal. We don't have yet a p-value that I think is convincing, because you have to say what was your original opportunity space. And the original opportunity space, depending on how you define it, could be only the stop variants that are seen in the genomes, or it could be all the, say, unequivocally functional categories like essential splice and non-synonymous, and so your p-value might be around 10 to the negative 5 or 10 to the negative 6. And this is the best that we're getting so far for these variants. But now, of course, the ones that still look good, we will carry out into further genotyping, and the hope is that this chain will eventually identify sufficient evidence for association. And I think that that really is going to be one of the primary designs for discovery. Until we can get to the point of really just doing tens of thousands of genomes, I think that this will be a primary engine of discovery, taking these out into large enough samples. The other kind of comment I'd like to make is that one of the beautiful things I think about human genetics is that it really is supposed to tell you about things that you didn't already know about. And for that reason, we have taken a very strong view that you really don't pick genes. You let the genetics speak for itself. You take what genes come out and whatever they are. So that is very much our philosophy. But even so, I have to say that even from that committed human genetics perspective, some of the genes that you end up with are really pretty crummy from the perspective of neuropsychiatric disease. This one in particular, I don't know if anyone can read that, it turns out that that gene is one of the principal components of hair. And so it really doesn't make it high on anybody's list for either epilepsy or schizophrenia risk factors. However, if you look at the mouse brain atlas, it turns out that this is indeed expressed in the mouse brain. I haven't a clue what that means, but I actually would not being facetious, I would take the view that you really can't pick and choose. And once you can't pick and choose, you really just do the genetics and you see where it takes you. And sometimes it shows you something that you were not expecting. So that's an example of following up classes of genetic variants. And we have also been thinking about this idea of zeroing in on particular extremes. And I'll tell you just about a few that we're focusing on. We're particularly interested in the idea that you can use drug response to zero in on a more homogeneous subset of patients. And so in both epilepsy and in schizophrenia, we're looking at particular subgroups based upon responsiveness. In epilepsy, there's a very well-known phenomenon of distinction between patients with juvenile myoclonic epilepsy, or JME, whereby the majority of them respond very, very well to valproic acid, but a minority of them do not have their seizures controlled. And so we have tapped into our clinical cohorts in epilepsy to try to build a cohort of refractory epilepsy. We've done something similar in schizophrenia, and I'll tell you about two others at the end. So first, on the JME side, as I said, a fraction of individuals do not have their seizures well controlled, and we combined selection of those individuals from sporadic families with, again, a multiplex family study being done through NINDS funding, and combining both what we see in families with what we see for sporadic cases. We have been sequencing JME patients, and then again, all the variants that show either presence in multiple affected in families or enrichment in the refractory JME patients, we're carrying into follow-up genotyping in larger cohorts. So here's an example of some of the variants that have been taken into follow-up genotyping, and this can be either based upon enrichment in the cases, either on allelic or a recessive model, or in regions that show linkage for JME, or in families. And that follow-up genotyping for 9 variants of interest showed that 8 of them were not involved at all, so we could rule them out. And one of them had the association evidence strengthen quite sharply in the follow-up genotyping, and so it is possible that a non-synonymous variant in that gene may in fact be a risk factor for JME. I again do not know yet whether that's true. I'll need yet more follow-up genotyping for that, but I think we are probably on a pathway that will eventually work. And I guess the one thing that I really, really like about this candidate gene is that it has absolutely nothing to do with hair. So this is a transporter which transports glutamate, probably. So it is an okay candidate gene. But in any event, like I said, you follow where the genetic association takes you. Where are we now in terms of p-values? Well, if you just throw all the cases together, you actually, your p-value is fine, 10 to the negative 7. That is actually not okay to do because we know very well that population structure can inflate these association statistics. And so you don't get to ignore that just because you're looking at sequence data. So if you then take these variants and do a proper control for stratification in the same kind of way as we used to do for GWAS, only in individuals of a certain continental ancestry, so in our case we did it for European ancestry, p-value is a bit lower and is a bit higher, and we're not quite to where we need to be, but I think we actually for that one may be moving in that direction. And we are doing a very similar study for treatment-resistant schizophrenia. This is tapping into a very long-term set of cohorts that Herb Meltzer has been developing with very well-characterized treatment-resistant schizophrenia, and it is also, again, tapping into cohorts that are being developed with ARA funding at Duke, and also a collaborator of ours at McLean Hospital, Deborah Levy. So these sources are described here. And the design is that we will be sequencing as many of the treatment-resistant patients as we can find, both from families and sporadics again. It's a combination of whole exome and whole genome, and then we're taking the variants of interest into follow-up genotyping, the exact same design as before. And so far we have about 100 of those complete. The last two relative extreme traits, or very extreme traits I'm going to talk about, are drug-induced liver injury and resistance to infection by HIV. The drug-induced liver injury example, I think, is particularly interesting in that this represents a very dramatic extreme from the population. So for some drugs, it may be one of 10,000, 50,000 or fewer exposures that can lead to serious liver injury. And so we're talking about a real minority of the population. And it's entirely possible that there are, in those cases, extremely strong genetic control that would be picked up in relatively small sample sizes. And if you then found the genetic control, it would make a real immediate difference because you could then avoid the drug in those individuals that are susceptible. And there are, in fact, already indications of exactly that kind of genetic control for drug-induced liver injury, for example, with flucloxacillin having such a strong dependence on HLA-B 5701. So this has really encouraged us to pursue a sequencing effort in drug-induced liver injury, and this is being led by Tom Urban in the group. And we've been able to establish a collaboration with a really exceptional resource that's been put together by the Dillon Consortium, the Drug-Induced Liver Injury Network. And this is participating sites, and this is a consortium effort that is headed by Paul Watkins. And we are now pursuing a variety of sequencing studies in partnership with them. They have a total of 1,000 patients with carefully confirmed drug-induced liver injury where they really have a high degree of confidence that they know the drug that's responsible. There are a modest number of drugs where there are a largish number of patients that are affected, and then there's a bunch of drugs with only a few individuals. And so the design that we've been following is to sequence all the cases where there are more than 5 cases for a drug, and then carry out those analyses first. And then we may, in fact, go back and even sequence where there's only a few of each, fewer than 5 for each drug, to look for common risk factors across the drugs. And so far, we've done sequencing for valproic acid and also for isoniazid. Very small numbers, and so we don't have anything definitive yet, although there is actually, just saw a very interesting possibility with valproic acid. Finally, I want to turn to perhaps the area where I think there is the most promise for near-term discovery, and maybe already a discovery, and that is looking at the genetic control of who can and who cannot be infected with HIV. This work is being led by Kim Pilak, and it capitalizes on a cohort of individuals that were exposed between 79 and 82 to either known or suspected contaminated infusion factor as part of their treatment for hemophilia. This cohort is being assembled by CHAVI, an NIH-funded network to study the immunology relevant to developing an HIV vaccine. There are about 500 individuals that have been assembled that had infusions during that time and did not become HIV positive, even though the majority of people did. I should also say that we are making use in this effort of another cohort that Jim Geddert assembled at the NCI many years before the CHAVI effort. And that cohort, in fact, probably has slightly higher overall exposure rates than the CHAVI one, because Jim put a great deal of effort into selecting the very most exposed individuals. So those individuals are available to mine the genome for variants that confer resistance. And what I'd like to emphasize here is that we know of one genetic factor that certainly confers protection against HIV infection, and that's homozygosity for CCR5 deletion. That's an obligate co-receptor for HIV, and so if you don't make the appropriate form of the co-receptor, then most forms of HIV can infect. But we also know that there are other people, probably a majority of people, that don't have that, that can't be infected, and that they almost certainly have something yet to be found genetically. So that's what we're trying to do. So that the goal is to find these protective mechanisms. And I should also say that one of the reasons that we're particularly interested in doing that is the idea that if the strategy for controlling HIV were to turn more towards prophylaxis, I'm not saying that it will, but if it were to, then it becomes relatively more important to have more drugs and more options for prophylactic treatment. And one route to that is finding ways to have drugs that act on the host. And so we need to find more natural variants that confer resistance, which would give us clues to drug development. In any event, So far, we have sequenced, in fact, 50 individuals that were exposed from 79 to 82 and were not HIV positive, and that's 48 at the time of the analysis. We've now done 50, and we're comparing these either to whole genome sequence that we generated in the lab that are not exposed, or whole exome sequence, and looking for variants that are enriched. And when we did that, we actually found some quite striking associations immediately just in the sequencing data. And so here are a few of the most interesting associations that emerge from the sequence data. So we have variants that, based on the sequence data, achieve experiment-wide significance quite readily. And in 2 cases, these variants are in genes that actually came up in an RNAi screen for gene products that influence in vitro replication of HIV. Now, as I said before, you don't know anything until you follow up genotype, so I'm not claiming this is real. They look real from the sequencing. We need to follow up genotype. We'll see what happens. But this really actually looks very, very encouraging at this stage, and we're just now in the process of following up these variants and other variants. One point I really do want to emphasize is that in doing any of this kind of work, it's absolutely critical to have really good follow-up resources to confirm the effect of the variants, as I said earlier. And in our case for HIV, we really are in a terrific position because of all the efforts of cohort building that have been going on. And so the way that we'll be able to follow this up is to genotype variants of interest in further exposed individuals that did not become infected. So as I said, we have around 500 of the exposed hemophilia individuals. We also have individuals that are exposed through sexual contact. We also can look at the variants in the general population, and the expectation, of course, is that there will be an enrichment in the exposed but not infected individuals relative to the general population. But finally, we can also look at putatively protective variants in HIV positive cohorts. And there, of course, we expect the variant to be depleted. And so this is already, just in our own group, the numbers that we have available for this follow-up effort. This is, in fact, over 500. So I think we really are here in a position to be able to confirm anything that we find, and the sequencing really just has to give us those options. And so one comment I'd make here is that I think this really illustrates the importance in discovery genetics of groups really working together, because I think what we don't want to see is one group trying to get everything that you possibly can out of sequencing 150 individuals but not having many for follow-up, another with 200, and so on. As far as is possible and reasonable, this work does work best when it's pursued together, and you can actually have this kind of a size of follow-up cohorts for confirmation that will also reduce the number of false positives that are contributed to the literature. So that's really what I wanted to talk about. I'd just like to end with some comments about sort of where I see things going. One of the things I think that's really critical to appreciate in the way genetics is moving, the direction genetics is moving, is that when we find variants that have a relatively high impact on the trait, it's really important to be able to look at the subject that carries it and also the subject's family to really work out exactly what that variant does. So follow-up phenotyping for carriers becomes relatively more important. Now this, of course, was very common in Mendelian genetics, very much less so in the area of genome-wide association study. Now that we're just moving towards sequencing genomes, we need to go right back in that direction. And insofar as possible, we actually want to be sequencing for discovery for common diseases, individuals that are still available for further evaluation, and that's really important. So we have a kind of return to classical human genetics. Another really absolutely critical question that we're going to have to face as early as possible is the extent to which causal mutations that we identify cluster into common pathways. Because one of the concerns about how to personalize medicine, if a lot of the action are rare variants, is that everybody's got a different cause of their disease. And to a certain degree in some diseases, that will happen to a certain degree. But it's entirely possible that the different mutations that individuals have will be clusterable into common pathways of some kind that have treatment implications, and that will be a really important area of focus. And the final comment is that to the extent that we start identifying relatively high impact rare variants, I think it actually will be possible to make more meaningful distinctions amongst individuals in terms of the risks that they face for disease, and that really might start to have implications for prophylaxis. Think of one example right away. You could imagine that if you could with more certainty predict which individuals might go on to develop schizophrenia, that you actually might start treatment, or at least evaluate treatment options even before the first episode. So I think we're actually in a very exciting time. I think that there will be a tremendous amount of discovery with sequencing, but I think we really have to be extremely careful about exactly how we deploy it to make sure we really get it as right as we can moving forward. So that's where I wanted to end, and just really thank the people that have been working with me on the various projects. I think that I mentioned, I hope, most of them as I went through, but the others are indicated on the slide. And thank you very much for your attention.
Well, we have time for about 5 minutes of questions, and then we can all join us for a reception at 4 o'clock. But there's certainly time for questions from the audience. I'll start with the first one. So the one thing I wasn't quite sure I followed, when you were talking about one of the stories about schizophrenia, you said you had data from next generation platforms and even Sanger-based sequence data, and it seemed like you still were unclear what the truth was, and I didn't quite follow that.
So, yeah, so for this one variant in DRD5, it turns out that the next generation sequence data is a bit messy, and Sanger is a bit messy.
So is it just part of the genome, part of region, where it's just hard to amplify?
It's just hard. It's actually hard to sequence it cleanly. There's homologous sequence, and I think we'll eventually get a clean enough sequence, but right now we don't. And it really just, I mean, I think just does emphasize that in the GWAS era, we were focusing on variants that we'd learned how to type well, and now we're dealing with a lot of variants that we haven't yet learned how to type well, and that you have to actually work through that.
And did a genotyping chip get you an accurate call at that position?
So that there's no, because that variant is not known to anybody.
So
it's not on any chip at all.
Got it.
Yeah.
Okay. Questions? It's not going to a microphone. No questions from the audience? If not, we can start the reception early. So please join us. There'll be a reception just a few yards down here out in the hall. Thank you very much.